Alzheimer's, Parkinson's, and other neurodegenerative diseases are associated with the inflammatory process in the brain. German researchers have succeeded in identifying a group of proteins in liquor that can provide information about the inflammatory process. As called biomarker, protein can help to better understand the disease process in the future and to test the effects of potential drugs on brain inflammation. The research team, led by Stefan Käser and Professor Dr. Matthias Jucker at the Hetty Institute for Clinical Brain Research and the University of Tübingen, in collaboration with Professor Dr. Stefan Lichtenthaler from the Munich website of the German Neurodegenerative Disease, has published it from German Munich, has now published Neurodegenerative Disease, has now published Germany, has published Neurodegenerative Diseases, has now published Germany, has published German Neurodegenerative Studies in the PNAS journal. The inflammatory process in the brain is common in Alzheimer's and Parkinson's disease, explained Käser, who led the research. In this process, what is called microglial cells play an important role. Microglia usually protects our brain from pathogens and harmful substances. In the case of neurodegenerative disease, chronic cells are active and release the substances themselves, said Käser. We suspect that this reaction initially had a positive effect on the course of the disease, and then turned into a negative impact later on. To learn about dynamic inflammatory reactions in the brain, the Tübingen research team has now sought the possibility of molecular biomarkers. Biomarkes is a substance that presence or changes in concentration in the body indicates the disease process. They can be measured in the blood, urine or other body fluids, and is an important tool for medical diagnostics, or to monitor the course of the disease. In this study, neurologists analyzed cerebrospinal fluids from mice, which showed the pathology of the characteristics of Alzheimer's disease or Parkinson's disease. Using sophisticated measurement technology, we can measure more than 600 proteins simultaneously in only 2 microliters of small liquids. We found that the concentration of 25 proteins was changed in both mouse models compared to healthy animals at the same age. Stefan Käser. Amazingly, most of these proteins come from or are related to microglial cells, the nerve biome continued. Almost all of them can also be found in human cerebrospinal fluid and changed in Alzheimer's patients. The concentration of protein that changes can reflect different